Hello, I'm Maria Williams with the Collaborative on Health and the Environment. CHE enjoys bringing you the latest environmental health science through our partnership calls and webinars, news feeds, publications, and social media. I would like to welcome everyone to today's CHE EDC Strategies Partnership Webinar, which is titled Successive Generations of Estrogenic Exposure in Male Reproductive Health. Our moderator today is Charles Patton, Director of Commonwealth's Biomonitoring Resource Center. We will leave time following the presentation for a brief Q&A session. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentation. After the presentation, our moderator will read questions out for our presenter to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on the webinar right now is muted with the exception of our moderator and our speaker. This webinar is scheduled to last for 30 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Charles.
Thanks a lot, Maria, for that introduction to the call, and I'm extremely delighted to present Tegan Horan presentation today. It's compelling research and has great significance as we all are observing the downward spiral in sperm count around the globe and increasing rates of infertility. Tegan Horan is a PhD candidate and trainee in Patricia Hunt's laboratory in the School of Molecular Bioscience at Washington State University. The complex biocultural origins of sex and gender are at the heart of her research interests, and to that end, she has completed a master's degree in genetics from the University of Adelaide, where she studied the evolution of spermatogenesis in the platypus, and a master's degree in anthropology from Washington State University. In this current research she's going to be describing for us, she's used a sensitive and quantitative marker of germline development, meiotic recombination, to test the effect of successive generations of exposures on the testes. The results of her research indicate that continued exposures to estrogenic chemicals may be a leading cause of infertility. She has a lot to tell us today, and I want to jump right into her presentation and look forward to your questions at the end of her presentation. So Tegan, please go ahead. Thank you.
Okay. Thank you very much, and thank you for inviting me to discuss some of my recent research. As Charles said, today I'll be talking to you about the effects of multiple generations of estrogenic exposure on male reproductive health. Now, whoa. Oh no, there we go. Now, infertility is a growing concern. Today, about 1 in 10 couples are incapable of conceiving after 2 years of trying, and there are both male and female causes to infertility. However, for many couples, it's unclear where that cause of infertility actually is, whether it's on the male side or the female side. In general, more research has been done on the female origins of infertility, and only recently have we started to pay a lot more attention to the male side of this equation. Now, many of you may have noticed recently there's been a story circulating in the news about a precipitous drop in sperm production. And these headlines in the popular press are all in response to a recent paper by Swan and colleagues. In Swan's paper, they reported that there is a 59% decline in human males' sperm counts within Western nations over the last 40 years. Now, this paper is a comprehensive, systematic review of over 180 studies ranging from 1973 to about 2011. Now, while this decline in sperm counts is indeed very shocking, there have been previous reports over the last couple decades that have had very similar claims. Those reports had been met at the time with a lot of controversy, largely because a lot of data just wasn't available for sperm counts from 3 generations ago. However, with this new paper, the data has become so thorough, so comprehensive, that now even the skeptics are starting to come around and realize that, yes, sperm counts are indeed declining, at least in Western nations. Now, importantly, sperm counts are not the only adverse male reproductive phenotype that is on the rise. Indeed, there are increases in reproductive tract disorders, including hypospadias and cryptorchidism. And there are also increases in the incidences of testicular germ cell cancers. Taken together, these different disorders are termed testicular dysgenesis syndrome, or TDS. It's widely thought that TDS is caused by developmental exposure to estrogens. Now, unfortunately, there are numerous sources of estrogenic chemicals in our everyday life, from plastics to drugs to household care products, soy-based diets, growth-promoting chemicals, feminine care products, and pesticides. In fact, these chemicals are so ubiquitous in Western nations that it's basically impossible to avoid any exposure. There have been several epidemiological studies on chemical exposures in humans that suggest that these chemicals can be related to the increase in TDS over the years. However, it's important to remember that these kinds of studies look at chemical levels in human adults. Whereas experimental studies that have used model organisms, mostly rodents, have been able to actually target developmental windows, and they have provided very compelling evidence that strongly supports that exposure to estrogen-like chemicals can cause TDS. Now, our laboratory's focus has been on mistakes that occur in the production of eggs or sperm. We know that these kinds of mistakes regularly happen in the female. For instance, aging is a major influence in errors egg production. There are remarkable ways, though, however, that both the female and the male respond to mistakes during gametogenesis. So, for instance, in the female, errors that lead to gross chromosomal abnormalities, such as this image where the chromosomes are kind of aligning themselves helter-skelter, this would still create an egg, but would result ultimately in embryonic lethality. Such errors in the male, however, result in cell death well before a mature sperm can actually be formed. Now, our lab kind of stumbled into the endocrine disruptor field by accident. We had noticed that several of our control female mice were showing these really striking chromosomal abnormalities, and we were able to trace it back to damaged mouse cages. These cages had actually been leaching bisphenol A, or BPA, an estrogenic chemical, and that was causing these chromosomal abnormalities in the female. Now, we've done subsequent tests in the male and found that indeed BPA exposure causes similar errors in the male that, as I said again, result in cell death. Now, within the last 10 years or so, there's been growing evidence that exposure to estrogenic chemicals like BPA can be passed on to subsequent unexposed generations. And so, for an example, let's think back to changes in sperm counts. Say that we expose a newborn mouse to ethinylestradiol, here represented by EE. It's important to note that while this exposure is affecting this individual mouse, it's also impacting his developing germline. Those germ cells, once this mouse reaches sexual maturity, can go on to co-constitute the second generation. And so when these two generations, the first and second, show reduced sperm counts, that would be the result of that direct exposure. They've both received a direct exposure to estrogen. The first generation, because it was orally administered, second generation through their father's germline. Now, if those reductions in sperm counts persist into the third generation, which importantly has not been exposed, we can say that that reduction in sperm count is the result of an ancestral exposure. Basically, that initial exposure that this third generation male received from his grandfather is an inherited effect. This paradigm is basically what's being used in transgenerational studies, wherein transgenerational means that the effect is observed in that third unexposed generation. Now, these kinds of experiments are very, very important, particularly for understanding the, for understanding the long-term effects of chemical exposures. However, they're not particularly representative of the human situation, in which for the last several decades, we have been continually exposed to various estrogenic substances. And because humans are continually exposed, our lab decided to ask a slightly different question, namely, what happens if we expose every single generation? And so we started with this exposed male, and then we exposed his sons and his grandsons. This basically gave us two lineages of mice, one in which only one generation was exposed, and the other in which all three generations were exposed. Now, we were surprised with some of the effects that we saw as a result of multiple generations of exposure. Among the first that we were able to identify were malformations of the male reproductive tract. So here I've actually got an image of what the mouse reproductive tract looks like in context. In the center, we've got the testis. Above the testis is the epididymis. This is the organ where sperm undergo maturation. And then on the bottom, we have the vas deferens. This is a long tubule that delivers sperm. Now, in our study, we saw a normal vas deferens. Here we see that typical long tubule shape, but we also saw several defects. Basically, we saw a convolution of the vas deferens along its entire length in a phenotype that I termed kinked. We also, however, surprisingly saw a more severe version of this phenotype in which the vas began to curl in upon itself. This is a phenotype that I then termed collapsed. When we look at the incidence of these phenotypes across generations, what we found was rather remarkable. The founder males, that first generation, 2 out of 3 of these founders had the kinked phenotype. They were mildly kinked. And in the second generation, we started to see the appearance of this more severe collapsed phenotype. However, by the third generation, virtually every single male was showing some kind of abnormal phenotype. And indeed, the slight majority were the more severe collapsed phenotype. In addition to this, in that third generation, we shockingly found a novel defect in males. These were fibrotic testes. And so here again, we've got that typical reproductive tract versus this, a more severe version of this fibrotic testis phenotype. So here we've got the epididymis, the vas deferens, and in the center is the testis. What's striking here is that the reproductive tract is actually fused with the testis. Now, these males, these testes are characterized by an expansion of fibrous tissue inside the testis that completely shuts down spermatogenesis. Basically, these males are sterile. Now, again, this phenotype was only found in the third generation, only after multiple generations of estrogenic exposure. Now, notably, other researchers have reported similar phenotypes to this and the malformation of the reproductive tract as a response to estrogenic exposure. However, those studies are using a much, much higher dose of chemical than what we are using. And so what we think is happening here is that we are increasing the sensitivity of males to further exposure as a result of the multiple generations of exposure. Now, going a little bit further, I have to take a step back and explain a little bit of what we do in our lab. We're very focused on the nuances, the molecular happenings of spermatogenesis and oogenesis. And in making a sperm or an egg, a lot of very important, very remarkable things must happen. We have to basically take the chromosomes that you inherit from your dad and your mom and segregate them into different cells evenly. So first, your mom chromosome and your dad chromosome have to meet up. They have to pair. And then they actually break and physically exchange parts of their DNA. Now, this has two really important functions. One, it ensures that the chromosomes that we get are not identical to either those of our mom or our dad. However, this process of forming those breaks also physically holds the mom and dad chromosome together, ensuring that they segregate properly into daughter cells so that we get appropriate chromosome numbers in sperm and egg cells. So in this picture that I've just put up, those red lines represent a protein scaffold that forms between the mom and dad chromosome, whereas the green dots represent these exchanges that are physically linking these two chromosomes together. Now, noted by the white arrow is a chromosome pair that has failed to form an exchange. This is actually a fatal error in males because there is a very robust mechanism that detects chromosomes that are no longer paired and kills the cell. So this cell would die before it could become a mature sperm. When we look at the incidence of cells with these failure to form exchanges, we see about 10% of cells in the founder have this fatal error. And this is pretty much on par with what we've seen in previous studies from our lab. Going out to the second generation, we see a slight but non-significant increase. But remarkably, in that third generation, we see a huge increase, up to about 35% of cells containing this fatal error. So what we're really seeing here is that this third generation of continual exposure is really critical. First, we're seeing a massive increase in fatal errors during spermatogenesis. These fatal errors should then translate into a reduction in sperm production. We're also seeing an increase in reproductive tract defects. And we're seeing this novel, severe testicular fibrotic phenotype that's resulting in infertility in this third generation. Now, let's think back to what Swan's paper is saying, that we are seeing this massive decline in sperm counts over 40 years. Now, if estrogen exposures are driving the observed reduction in sperm production, certainly the data seems to imply that they are, then our findings seem to suggest that human populations may be growing more sensitive to these exposures. The volume and the diversity of estrogenic chemicals has only been increasing in Western nations since the 1940s and 50s, such that now we are approaching the third generation of continual exposure to estrogenic chemicals. Looking forward, we have to ask, are we on the cusp of an infertility crisis? Are we on the road to infertility? Or is there still some way, some time to recover from these continual exposures? It's also important to remember going forward that there's a ubiquity of sources of estrogenic exposure. And we can take, for example, bisphenol A. Now, up until fairly recently, a lot of products, including Nalgene water bottles, had BPA. Now, we see them all with this sticker that says no BPA, they're BPA free. So problem solved, right? Probably not, as these companies have just replaced BPA with several other bisphenol analogs that are almost structurally identical to BPA. Now, early studies are starting to suggest that these so-called safe replacements may actually be just as harmful as BPA, possibly even more harmful. It's also important to note that these bisphenol chemicals are not the only source of estrogenic contamination in our lives. And indeed, not all chemical exposures are even estrogenic in nature. We're also exposed to a lot of anti-androgenic chemicals, and some chemicals have mixed modes of action. So moving forward is going to be really critical to build models that take into account this mixture as well as the continual exposure that humans are experiencing. And so with that, I'd like to thank the members of the Hasselt and the Hunt Lab who have helped me both in the data collection and practicing this presentation. I also like to thank several members of the Griswold Lab who helped with data collection. And of course, our research here is funded through the NIH. And I'll take any questions now.
Okay, thanks very much for this compelling presentation, Tegan. That is, sounds the alarm bell, I think, for many of us. We have some questions coming up on from our participants. And the first one is, were the females unexposed in all 3 generations?
Yeah, the females were not exposed. So what we were using was an outbred mouse strain, and all the females were unexposed. And with each generation, we use completely different mice so as to keep our mouse population as outbred as possible.
Okay, and the exposure levels that you used for the 3rd generation, are those equivalent to exposures that humans are being exposed to of bisphenol A, for example, in the environment?
So we used ethinyl estradiol as our chemical, and we use it for a bunch of different reasons. One, because we've used it generally as a positive control in our bisphenol studies, but also because we wanted to have an effect that would be robust enough to be trackable, but not so robust as to completely wipe them out within 1 generation. So we actually used an exposure of 0.25 nanograms per gram body weight, which is roughly equivalent to human exposure to ethinyl estradiol in birth control.
Okay. I'm just writing this down. So where's your okay. Have you replicated the increase in chromosomal irregularities in the 3rd generation exposed with E2 or other estrogenic chemicals?
The 3rd generation exposed with E2. So I'm a little confused as to what they mean specifically by replicated the increase. So we have seen this increase in exchangeless chromosomes even within a single generation with exposure to ethinyl estradiol and BPA in the male. I've also just completed a study looking at several bisphenol analogs, specifically BPS, BPF, BPAF, and diphenyl sulfone. And they also have that increase in exchangeless chromosomes.
Mm-hmm. What rates of exposure did you use?
We orally expose them using a pipette, and our chemicals are diluted in corn oil. Okay, okay.
And have you looked at the 4th or 5th generations?
Oh gosh, unfortunately we haven't. And if there's one regret we had from this experiment, it's that we didn't push it even further. And so we would really like to do that, but we did not for this project, unfortunately.
Okay. Would that be some area you might be going in subsequent research as you move forward on this?
Possibly. At the moment, we don't have any plans to do that. But I think at some point it's going to be necessary to see how far these effects go. Yeah. Yeah, this is really scary stuff. Okay, are there any more questions from our participants? Oh, I've got some coming up here. Somebody just says, and I want to share this with you, fascinating, groundbreaking research and beautiful slides. Thank you.
These slides are actually much more interesting and colorful and clear than some of the slides we've seen, so thanks for that. And the question is, what is your lab doing next?
Okay, so as I said, we've just started to wrap up an experiment on the effects of bisphenol analogs. And so we'll be writing that up pretty soon. That actually came as a result of some contamination that we had in the lab through damaged cages yet again. It never ends. And so that put a lot of our research on hold. We're just starting to get back up. And right now we're actually trying to figure out what specifically these exposures are doing. We've been able to track the exposure effect to what's called the spermatogonial stem cell, which is the cell that's responsible for the continued production of sperm throughout a man's lifetime. And we know that these exposures are specifically affecting that cell, but we don't know what it is. We suspect the change is epigenetic in nature. And so we're going to look to try to identify different chromosomal and transcriptomic changes in that cell type. And then we're going to, I guess from there, try to figure out how those changes are basically preserved from the spermatogonial stem cell all the way through spermatogenesis, because there are several cell divisions that occur in spermatogenesis. And so, in effect, at the very top of that chain, being present throughout is pretty striking.
That is really interesting. Well, okay, well, I look forward to seeing how that goes. As some of our participants may have questions after the call, would you be willing to answer them if we forward them on to you and then putting your responses and the question up on our website?
Sure, absolutely.
That would be great. Oh, from Ricardo Mauro asks, what about fertility parameters on those third generation males? Were they infertile or subfertile?
Okay, so yeah, this is actually important. So I kind of forgot to mention that fibrotic phenotype that we observed, that occurred in 20% of those males. So like 1 in 5. And we suspect that those males were generally sterile because, again, we could not get any evidence of spermatogenesis occurring in those testes at all. We could not get sperm out of them at all. As far as general fertility parameters in the third generation males, we don't have those because we didn't pair them to breed at that point. We had decided to just stop at the third generation. And so we don't have information about litter numbers or sizes and or how many pairings with females, unfortunately.
Okay. Well, but you've done incredibly provocative research. It raises a lot of questions and points to some answers that I think are quite alarming. And I want to thank you for your research and this excellent presentation. And as other questions come in, participants in the call, don't hesitate to forward them to me at commonweal.spatton@igc.org, and we'll make sure they get to Tegan and get the response up on our website. So thanks again, Tegan, for an excellent presentation, and I look forward to following your career and further research. So you're doing such incredibly important work. So thank you very, very much. Very deeply appreciated.
Yeah, thank you.
Okay. Maria, do you want to go ahead and formally close the call, please?
Yes, thank you, Cheryl. We are approaching the end of our webinar today. A video recording will be available on the CHE website later today, and tomorrow you will receive an email containing a link to the recording. CHE's next webinar will take place Wednesday, October 4th, and is titled Chemicals in Consumer Products, Exposure Science at the Forefront of Regulation. This is the first webinar in our new series, 20 Pioneers Under 40 in Environmental Public Health. You can RSVP through CHE's website, healthandenvironment.org. The next webinar from the CHE EDC Strategies Partnership will be held on Wednesday, October 18th. Details will be available on our website soon. If you are new to CHE and would like to learn more about becoming a CHE partner, please visit the Join Now page of the CHE website. Additionally, if you appreciate these webinars, bringing you the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank our speaker, Tegan Horan, for taking the time to present today, and Charles for her excellent moderation. Thank you very much for joining us, and have a great day.
Thanks, everyone, for joining in, and bye.
Thank you.
Bye, Tegan.
Thank you. Bye.